I want to thank the organizing committee for inviting me and putting together this great program. Now, I want to start off with a little bit of a denial. Contrary to what John Wood's been banding about, I was actually not the first person to do pain research. And in fact, I was digging through the literature to find out who that was, and it turns out that was somebody working not far from here, Edgar Adrian, and he was taking advantage of a breakthrough in our ability to monitor electrical activity in single neurons, and that was the cathode ray tube, which before that they had to use a string galvanometer. I know I'm using terminology you never heard of, but you can look it up on Google and Wikipedia. So I want to point out that this was 1928 when he was doing these recordings, and he got a Nobel Prize in 1932. He was very clear that the pain message was encoded in the firing frequency of a peripheral nerve, a nociceptor, and he knew that they were slowly conducting fibers. So I would say that this really began the neurobiology of pain. And a lot of what we know is built on the idea that what we feel is encoded in the frequency of firing of neurons, where primary afferent, second-order cells. Building on this work, we now know just about everything we need to know about pain. I'm not sure why everybody's here in the auditorium, unless it's just for nostalgia. But I'm not going to review this. You can find this in the textbook of pain. We do know about the neurotransmitters. We know the fibers. We know the CNS structures. And because of the development of functional imaging, we actually know a lot about the higher cortical mechanisms that are involved in the mediation of sensation. I would say, and I have skilled and knowledgeable people here who can correct me if I'm wrong, that we actually know more about pain sensation than we do about almost any other function of the nervous system. Anybody want to dispute that? In this room? John, okay. I knew that there was somebody in the audience that would be making trouble for me. Okay, so we can go from receptor activation to a perceptual event, and so we can explain pain, pretty much. And the psychophysics of it's very elegant. However, this meeting is about chronic pain, and chronic pain is kind of different and probably involves different circuitry. And what I'm going to propose today is not the only explanation, but it is kind of a provocative point of view. And that point of view is that the development of pain, as we know it, chronic pain, is partly a normal response of the central nervous system to a persistent nociceptive input. So it's a little bit different. And we know this, and everybody that's seen a lot of patients with chronic pain knows that there's a dissociation between what we see in our laboratory tests, in our imaging, in our conduction studies, and what the patient reports as the severity of the pain. So there's a dissociation. That's not true of the psychophysics of acute pain, where the pain intensity is lawfully correlated with the stimulus intensity. The other thing that these patients manifest is psychological comorbidities, anxiety and depression. But the thing I want to really focus on today is the role of expectation. And I'm going to talk about what it is, how it works, and how it influences pain intensity. And it's pretty clear that it does. This is a paper that was recently published in Pain, where they just looked at 2,000 patients who were entering treatment for chronic pain. And these were a variety, this is a multi-center study, so it was a variety of different pain centers. And they looked at they evaluated the patients when they entered treatment, and then they followed them up for a year or more later. And what they found was the single biggest factor that correlated with how well a person did in their treatment for chronic pain, whatever the treatment was what they expected would happen when they began treatment. So that's basically what this slide shows. So this is change in pain intensity over the course of treatment, and you can see that's highly correlated with what they expected when treatment started. Okay. So this is a study that John Keltner did when he was a postdoc with me, and it was really the first functional imaging study that I did or was involved in. And we were actually looking into this issue of expectation. And what we did was we gave subjects, these are normal volunteers, and we gave them two different temperatures, and these were calibrated for temperatures, approximately 47 degrees or 48 degrees. These are both super threshold for pain sensation, and the heat stimulus was delivered to the forearm. And when they got the high intensity stimulus, they saw this red thing that was displayed on their monitors. And then with the low pain, they had the color's off. It was green, actually, but I show it here as yellow. So with the 47 degree, they got a cue that was associated with that. So they had two cues and two pain temperatures. One correlated with high, one correlated with low. And then we did this oddball experiment of switching the pain intensity, the actual stimulus temperature, with the opposite cue. And as you might imagine, and here's the data shown, so this is a visual analog scale where 10 is the worst pain ever. So you can see 48 degrees when you're expecting it is very close to the highest intensity. We're aiming to give everybody an 8 with the 48. However, if that same temperature, when it was paired with the low temperature cue, in other words, this one had been contingently paired with the 47 degrees, they reported a significantly lower pain intensity, right? And with the low pain intensity stimulus, the same thing. If it was paired with a high cue predictor, the pain was greater, right? So looks like expectation can strongly modify what you feel when an acute stimulus is applied. So expectation is not just involved in chronic pain, it's also a big part of acute pain. And that becomes important in the argument that I want to make this afternoon. So I have to confess that I got my PhD in a biology department. So I have a natural inclination to look at things in an evolutionary perspective. And one of the questions one might naturally ask is, what's the point of having these expectation effects, which are clearly distorting the clarity of the signal that's coming from your nociceptors? Why would you do that? Wouldn't you want to have a reliable signal that you could depend on, that gave you information that you could count on? Well, that's a very simplistic view of what the biology is. So what's the biological function of pain? Well, yeah, all right, it's to protect your tissues, right? So you want to withdraw your limb, to keep it from getting crushed. So that was sort of what everybody from Sherrington on said. It's tissue protective. So that's really simple. The more intense the stimulus, the greater the potential damage, so you want to get out of there very quickly. So that works for the psychophysics of acute pain. However, other things happen, right? When something painful occurs. It usually occurs after you did something, right? And if you did something and pain occurs, then you're going to be less likely to do it in the future. It's called punishment, right? So that's the teaching function of pain, and everybody uses it. There's also this motivational aspect, which has been included in the psychophysics as the sensory affective, the affective motivational aspect of pain. But motivation biologically has a completely different meaning. It's not a psychophysical construct. It means get out of there or do something, right? If you're hungry, you want to eat. If you're thirsty, you want to drink. If you have pain, you want it to stop. So it's a motivation. So you've got these two aspects. These are what I call the biological meaning of pain. And I think if you understand that, then you can understand the role of expectation, why it's powerful, and why it's biologically useful to distort the signal. Okay, so let's do so this comes from my background when I took a sabbatical from pain research for 10 years and started doing addiction work. Now there's a test that's used in the addiction field called place conditioning, which is now being used more and more in the pain field. But it really started off to see whether drugs, would be rewarding. So what it is, it's a 3-chamber device. You restrict the animal to one of the chambers. You do something, right? You give them cocaine, you give them morphine, or you give them a painful stimulus. The beautiful thing about this is you put them in the other chamber and you don't do whatever it was you did, right? And so then, after they've been trained for 3 or 4 sessions, you open up the doors and just see where does the rodent go, right? If it was unpleasant, the rodent's going to go out of the room where it received that experience. So that's what we call the teaching signal, and it has motivational value in and of itself. Okay, so what we showed, this was work that Josh Johansson did when he was in the lab, that if you make a lesion of the anterior cingulate cortex, then the animals will still withdraw from pain, but they don't learn that it's unpleasant, right? And the imaging people had told us that the anterior cingulate and the anterior insular cortex encode the aversive qualities of pain. So this was a rodent confirmation of that. This is quite straightforward. Okay, so let's talk about learning, and let's put pain in perspective. So if you have a cue, that cue is a predictor of what's going to happen. So for example, in that work that I showed you with John Keltner, if the red cue came on, they would expect a high temperature, right? So you're going to have a prediction that's either going to be a benefit or a cost, or in the greater scheme of things, it'll be a cost-benefit analysis driving behavior, right? Then you'll have an action, something will happen, okay? Then there'll be an outcome, right? And the outcome, if it's painful, means you won't do what you just did. The psychologists call that extinction. And it's a general term. I'm going to talk a little bit more about the complexities of it, but this is the basic idea. Predictions come first before anything happens. They use predictions to inform decisions about behavior. Then you're going to do a behavior. Then you're going to have an outcome. So this is typically when the painful experience occurs. However, the next time you're in this situation, what happened to you before is going to influence your decision about what you're going to do in the future, right? So it's very critical for understanding this talk that you remember when it is that the pain occurs and how the memory of that pain affects future behavior, because that's critical to the arguments I want to make. Okay, predictions inform decisions, reviewed that, and expectations are what I call the psychophysical equivalent of predictions. So this is the critical point. This is where this idea of expectation comes in. We have expectations because our brain needs to make predictions, because we need to have some idea about what the optimal thing is for us to do in any given situation. So expectations, critical part of our behavior. We're doing that all the time, every minute, right? And this morning when you got up, you made some predictions about what was going to happen if you took the right train, if you got to the right bus, if you went to the right lecture hall. Something good was going to happen. Now, if something bad happens, the next time you probably sleep through and forget about taking the train and forget about the meeting. Okay, so this we have, one aspect of pain is the pain. So you made a prediction about how bad the pain is going to be. If your outcome is exactly as you predicted, then there's no learning. If the outcome is different than you predicted, then there's going to be learning. If it's worse, then you're going to get extinction of the behavior. If it's the same, there won't be any effect on future behaviors. The other key, cute thing about pain is if you have it, there's the potential for it to be rewarding in a weird sort of way, and that is if, for example, the pain is less than you predicted, that's a reinforcer. That's a reward, right? It's the same as if something good happened. In fact, it is a good thing that happened. And I also want to say that a lot of people use the term negative reinforcement, referring to punishment. They are the opposite. Negative reinforcement is the relief of pain. Pain is a punishment. Now, I just want to get that clear so that no one can blame me if you make this mistake in the future. Okay, so pain relief is a reward, and that's going to become critical in the things that I want to explain. Okay, so now we have this, we had our pain transmission system, we had our nociceptors, we had our representation in the cortex, but now we've got these expectation effects. What do we know about how these expectation effects are implemented? Okay, well, it turns out that we have a modulatory system. It's running in a top-down fashion. This is work, John was involved in this, Alan Bassbaum, John Liebeskind, one of the founders of pain research, Dave Mayer, Huda Akil. They traced a circuit that goes from prefrontal cortex. Some of the same places that receive nociceptive input are sending connections down to the midbrain. There's a big input from the amygdala. That turns out to be important. The periaqueductal gray region connects to a site in the medulla, the rostroventromedial medulla, and this connects back to the dorsal horn. So as a word of reassurance to those people who are, how many people here work on dorsal horn and the spinal cord? How many people work on spinal cord? Okay, good. It's a good thing. I just wanted to reassure you that this whole brain actually is sitting there in service to the dorsal horn. So you can relax and sleep well tonight. Even though there's a lot of brain up there, it all boils down to what's happening in the dorsal horn. But don't tell Clifford Wolf I said that. Okay, so this turns out to be really cool. Okay, so we got this modulatory system, and there's been some work on placebo, and placebo is the expectation of relief or the prediction of relief. Okay, it turns out that during the expression of placebo analgesia, you get what looks like activation in many of these same sites, including the periaqueductal gray and the rostroventromedial medulla. And not only that, this is Falk Eipert's work. He did it with Christian Buchel. I think it was in Hamburg. But then they did something really pretty extraordinary. They were actually able to image the spinal cord. This is not trivial. But they're very reliable people. And so here was this functional imaging. So there's blood flow in the spinal cord. It looks like it's in the dorsal horn. And it correlates with the intensity of pain. And it was lower when there was a placebo response. Right? So what that suggests is, what I just said before, is that this whole modulatory system, the effects of expectation that involve cortical circuits, is actually sending this message down to the first central synapse in the spinal cord. Just as this information is entering the spinal cord, you've got your whole brain sitting on top of it, instructing it about, what that information is going to be used for. And this experiment I love. We have somebody in the audience that was involved in this, Kathy Bushnell, and this was done at the Dental Research Institute. One of my favorite experiments of all time. And no one's doing any work like this now. So what they were doing, incredibly, is they're recording from dorsal horn neurons in the nucleus caudalis. Did you know about this work, Peter? I'm asking Peter. Okay. Well, Peter's right there. You're gonna, So what they're doing, the monkeys are trained. They have a little thermode on their lip, and in order to get juice, in order to get a reward, they have to initiate a trial that delivers a thermal stimulus to their upper lip, and they have to hold it there long enough. If they don't hold it there long enough, they don't get any reward, and they want a reward, so they had to balance the behavior so that the animal would work despite the fact that there was a noxious stimulus being applied. Okay, so that's why it's a cool experiment. They made it work. And not only that, But some of the neurons that they were recording from in the nucleus caudalis, they identified as projecting to the contralateral thalamus. Give me a break. I mean, that's really tough to do. That's probably why they're not doing it anymore. But they did get this. Right, all right, funding. All right, yes, that's an issue. Okay, so what they showed was when you record from these neurons, like a lawful wide dynamic range dorsal horn neuron, it shows an increase in activity that correlates with stimulus intensity. That's great, okay? So these are the real thing. These are neurons you can believe in. But here's some stuff you're gonna have trouble with. These cells start to fire as soon as the primate pushes the lever to start the trial, right? So it's getting information about a predicted reward. So here you've got a wide dynamic range, nociceptive neuron in an awake behaving animal, and it's starting to respond to a motor stimulus that's predictive of getting a noxious input, right? And they showed some cells had inhibitions and some cells, started to fire when the light came on. So there's all kinds of information that's getting, from up here down to your spinal cord. Okay, so I think I've beaten that one into the ground. What I want to do is, go back a little bit and talk about how some of this might happen, and we're getting back to single unit recording. And this is neurons in several different sites, in the periaqueductal gray, in the cuneiformis, and in the medulla. There are populations of neurons that have opposite firing patterns. So this was a striking discovery when we started recording here. We first started recording in the medulla because these cells project directly to the spinal cord. Because our original thought was, oh, well, you noxious stimulus, you get negative feedback, these cells are going to inhibit pain. But then when we found that there were two kinds of cells, and their firing changed before the animal actually responded. So this bar is the beginning of a heat stimulus. These are lightly anesthetized rats, and we're looking at their tail flick. So the withdrawal that usually occurs around 44 degrees, which is similar to the pain threshold in people. The off cell stops firing about a half a second before the animal withdraws its tail. This other cell, the on cell, begins to fire about 200 to 300 milliseconds before the animal removes its tail. So what we thought, and we adduced evidence for it, which is, I would say, pretty good. These cells are inhibiting pain, and they have to shut off in order for the animal to respond. These cells facilitate pain, and when they turn on, they enhance the pain message, right? So the key point is pain control from brainstem to spinal cord is bidirectional. Pain modulation is bidirectional. There are two sets of neurons with opposite firing patterns. That turns out to be very important, so important that I'm going to not only say it and show it to you, but show it to you in extra cartoons, because I think it's really important to remember. So, and it really gives you a simple idea of how you go about controlling pain. Okay, so here's your pain transmission cell. Let's say it's a lamina 1 cell, or it could be a lamina 5 cell, it could be nociceptive specific, wide dynamic range. It's receiving nociceptive input. This is determined by stimulus intensity under acute situations, but it's subject to two different forms of descending control. So if nociceptive input comes in and this cell fires at 10 hertz, but now you said, oh, well, I don't really want to respond right now. I'm going to turn my off cells on. Now these are inhibited, and this has the same input, but now this is firing at 5 hertz. So you can see how nociceptive input is part of the equation for determining perceived stimulus intensity, but modulatory inputs may be of equal weight. And sometimes, depending on how close you are to pain threshold, they might be the greater contributor. So just remember that cartoon, because we'll come back to it. I want to switch gears a little bit and talk about my favorite protein, which is the mu-opioid receptor. And it's my favorite protein, even though I did not discover it or clone it. But if you make a mouse that doesn't have a mu-opioid receptor, you can load that mouse with morphine and really nothing happens. they don't get any pain relief, they don't get any sedation, they don't get any respiratory depression, they don't get any decrease in gut motility, nothing. It's like water. So this receptor does a whole lot, okay? It's recently been crystallized, and it turns out that there are other receptors that have high sequence homology and are often co-expressed with the mu receptor, but I'm not going to get into that. I just want to talk about some of the anatomy and circuitry that are involved in the expression of analgesia from morphine. All right, so one of the first people that systematically study this was Tony Yaksh. So John told me I needed to be somewhat historical in this talk, so I want to recognize some of the major contributors to the story. Tony Yaksh, definitely. He was the first person to show that there might be an argument from Jean-Marie, but he was the first person to show that you could get analgesia from directly applying opiates to the intrathecal space of the spinal cord. It makes sense because you got mu receptor there. You can inject it into the medulla, mu receptor, you can get analgesia. Mu receptor, PAG, hypothalamus, amygdala. All of these sites are sensitive if you locally inject morphine. Furthermore, if you give morphine systemically, you can block its effect by putting naloxone in any of these sites. So here you have a serially connected circuit that's connected by the mu receptor, and we now know, because of the pioneering work of Hughes and Kosterlitz, and published in '75, that endogenous opioids, this is tyrosine, methionine, enkephalin, and leucine enkephalin. These are what they found from pig brain, and they have the pharmacological properties of morphine. Okay, and if you do the immunocytochemistry, you can show they're everywhere where there's the mu receptor. All the places that are sensitive to the microinjection of morphine have endogenous opioids, and that makes all kinds of sense. It's just to show that the off cells, which I proposed were inhibitory to pain, they're activated by morphine. On cells, which facilitate, are inhibited by morphine. So it all works. So morphine will suppress pain in part by directly inhibiting the release of neurotransmitter, but also by turning on this descending system, acting at multiple sites. There are other sites too. Frank Parekhin's group recently showed that you can, with ongoing pain, you can put mu opioids in the anterior cingulate and get analgesia. So that's kind of cool. Here's another pioneering study that John did when he was a student in the lab. There were a couple of papers that were published, one from Pat Wall and one from Avram Goldstein, showing that naloxone didn't do anything to pain. And I remember walking down the hall with John with a very long face. I wasn't exactly in tears, but I was upset. We'd been doing all this work on the modulatory system. The endogenous opioids had been found, and naloxone didn't do anything. And these were two major figures in the field. So John said, well, they must have done it wrong. Remember that phrase? They must have done it wrong. Okay, so John upped the dose and in fact showed that naloxone gave a very clear hyperalgesia. So we could get back to work, right? So we could say, all right, it isn't just something that happens in rodents, it actually happens in people. So that was quite an important part in going forward. All right, so now we can get back to the question of, what normally engages this descending modulatory circuit, right? Well, we know it's expectation, but what's the circuitry by which expectation, either of increased pain or decreased pain, activates the circuit? How am I doing for time?
You've got 20 minutes.
20 minutes? Oh, perfect. Okay, good. I could slow down? Okay. All right.
I don't know how many slides.
I don't have enough slides. Oh.
You'll have more.
Just kidding. Okay. All right. Okay. All right. So then the question is, okay, when do you want to suppress this pain signal? What would be the biology of it? There are times when you need to ignore pain in order to get on with whatever else it is you're doing. Ignore a cause. Okay. So, and this gets back to, history. Pat Wall and this is a very, famous but rarely quoted statement that Pat made, and this gets right at the heart of the matter. And he said, the brain analyzes sensory input in terms of the possible action which would be appropriate. That's a perfect way to state it, right? And that's the answer. That's what we come around to when we're thinking about when do we want to implement this modulatory system. Okay. So decisions, right? So in order for decisions to make any sense, you have to have alternatives, right? If you're a tree, you don't have a lot of alternatives. You don't have to make a lot of decisions. You don't have to move anywhere. You don't have to do anything. But if you're an animal, especially a vertebrate, there's a lot of things you can do. And if you're a human being, there's a zillion things that you could do, right? And you have a lot of different motivations. These can all be present at the same time, right? So then you have to pick and choose. You have to optimize a behavioral strategy. You have to sequence, your behaviors in a way that makes sense because you're competing with everybody else in the room. Or at least that's how we in the United States conceptualize it. Everybody's competing with everybody. Okay, so let's go back to our place conditioning apparatus. And let's say that the rat has had a painful experience in this compartment, and its natural tendency would be to avoid going there. But let's make the rat's life complicated by putting some cheese in that room, and then we'll see what the rat does. Well, clearly, if you just pre-fed the animal with a lot of cheese, it's not going to go in there. But if it's hungry, it'll go in there, right? So one motivation's competing with another motivation. If you separately manipulate the motivations, then the outcomes will differ, and they're changing all the time. So, this is what I call the basic situation of life. Are you going to go for the reward, or are you going to avoid the pain? Are you risk averse, or are you a risk taker, right? So there are personality features involved, but luckily I won't be talking about personality in this talk. Okay, so this is another, what I call great experiment. So this was done in Albert Hertz's lab in Germany, and what they did was they just brought rats in and they put them on a hot plate. They didn't turn the hot plate on, don't have to worry. And then they would, day after day, they'd bring them in, put them on a hot plate, and they fed them on a hot plate, right? So they either fed them their regular lab chow, or they fed them a chocolate-covered cookie on the hot plate, right? Well, so you can imagine what would happen when the animals had been trained with the chocolate-covered cookie, right? They would stay on the hot plate twice as long as the rats that got their regular lab chow, and they were the ones that got the regular lab chow were no different from controls, that were never fed on the hot plate. So there's something about palatable food that makes rats willing to endure a certain level of pain. And this, I propose, is what this circuit's doing. It's all about keeping the rat on the hot plate in the expectation of sugar and chocolate. And I'm sure everyone can relate to that. Okay, so this is what I call the motivation decision model. You get a noxious stimulus in the presence of a conflicting drive. You do a cost-benefit computation, you make a decision, and if you want to stay and get the chocolate or the cheese, then you're going to suppress your pain pathway. If you're going to get release of endogenous opioids, you can get your off cell going. If, however, the decision is, I better get out of here, you're going to facilitate your pain pathway, turn your on cells on, and you'll escape faster, right? So if you decide to escape, you want to do it fast. If you decide to stay, then you really want to suppress the pain completely. And that's why the system's so powerful. Okay. So where's the decision made? This is the new part of the talk for John. And that is in the reward pathway, what used to be called the reward pathway. I think it's a misnomer, and I I think you'll see why as I get into this a bit. There's this area called the ventral tegmental area. It's in the midbrain. It's the source of dopamine neurons that project to the cortex, the amygdala, and an area called the nucleus accumbens. So rats will press a lever to electrically stimulate anywhere along this pathway. What's seldom reported is they will also press a lever to stop the stimulation if the experimenter turns it on. So it really isn't a reward pathway. It's a pathway that exerts bidirectional motivational control for the animal. This is just to show in coronal section in red as the medial forebrain bundle, which connects the ventral tegmental area with the nucleus accumbens. Okay. So let's go back to our functional imaging buddies and ask if there's a pain predictive cue, do you see activation of the circuit? The answer is yes. Pain predictive cues activate the human nucleus accumbens. That's what's shown here. It's the same area, or at least it overlaps significantly with the area of the nucleus accumbens that shows a correlation with reward predictive cues. So pain predictive cues and reward predictive cues both activate the nucleus accumbens. So I'm going to do a little audience participation here. Because this is a group that does pain research, probably doesn't use the word nucleus accumbens very often, I'd like everyone to just repeat after me, nucleus accumbens. Okay, that will, it's a didactic device, and you know, it'll help you remember my talk. Okay, so this is work from Knudsen down at Stanford, and he was showing Stanford undergraduates like a roulette wheel, and that would show different amounts of money that they had just won or lost, okay? And what they found was that once you got up above about $1 or so, the blood flow started to increase in the nucleus accumbens, and it correlated with how happy they felt. Pretty cool. Remember, overlapping significantly with area responding to pain predictive cues. Okay, so we did some work, and this is work Salim Nicola and Irene Yun, a graduate student in the lab, just inactivating the ventral tegmental area with baclofen. Turns out that that relatively selectively inhibits dopamine neurons in the ventral tegmental area. And if you do that, or you put a dopamine antagonist into the nucleus accumbens, animals stop responding to reward predictive cues. Right? So the dopamine pathway from the VTA to the nucleus accumbens promotes responding to reward predictive cues. And there are, as you might have guessed, because I'm talking about it, neurons in the nucleus accumbens that show a sudden increase in firing when the reward predictive cue comes on. They fire until the animal actually gets the reward. So these cells are probably driving the behavior of approach to reward, okay? You know, like if you inactivate the ventral tegmental area, take out the dopamine cells, the firing of these neurons goes away, concomitant with the loss of behavior. Okay, so that's pretty cool. It turns out, and now we're getting into little basal ganglia neuroanatomy, the nucleus accumbens has two major types of neurons in it. D1, dopamine D1 expressing neurons, otherwise known as direct pathway neurons. These neurons also have substance P and dynorphin, which is a kappa agonist. And the D2 cells, these project to an area called the ventral pallidum. I won't ask you to remember ventral pallidum, but just to remember that there are two different types of cells. The D2 cells express enkephalin, right? So there are these two types of cells in the nucleus accumbens, and it turns out that if you activate the D2s, animals stop what they're doing. If you activate the D1s, it promotes approach. So it's possible that the cells that I talked about that are excited by dopamine or require dopamine for excitation are D1. We don't have confirmation of that yet, but it's likely because dopamine excites D1 cells and inhibits D2 cells. So these would be the go cells, these would be the stop cells, okay? And these activate VTA dopamine neurons. So there's a little positive feedback. Then there was some interesting work from Matt Christie's lab in Australia where he was doing a CFOS study to look at activation of what cells in the nucleus accumbens are activated by morphine. As you might expect, it's the D2 cells that are activated by morphine. These are the stop cells. Okay. Did I get this? Yes. The D2s are green. The CFOS is actually not in the D2s. I got it a little wrong. So the D1 cells are activated. The approach cells are activated. And withdrawal, morphine withdrawal, if you give them naloxone, that activates the D2s. So the stop cells are activated by naloxone, and the go cells, the D1s, are activated by morphine. Okay. And then there's some recent work from Jim Surmeyer at Northwestern showing that in the presence of a tonic pain model, you get increased excitability in the D2s. And when you activate D2s using optogenetic approaches, so you can selectively activate channel rhodopsin in the D2 cells, you actually get hyperalgesia and allodynia. So it looks like there's bidirectional control coming out of the nucleus accumbens, and some of this depends on reward predictive cues. And Giancarlo Zubietta did a study, again, getting back to placebo, where he showed that placebo reactors show an increased release of endogenous opioids. This is a PET study using C11 carfentanil. That's a short half-life mu-selective opioid agonist. And you sort of, you get indirect evidence for release of endogenous opioids because they would be unlabeled and they'll displace the C11. So what he showed was evidence for release of endogenous opioids in the nucleus accumbens and the periaqueductal gray by the expectation of reward, by the expectation of pain relief. Okay. Furthermore, back to rodents, if you inject a mu agonist into the nucleus accumbens, you not only get analgesia, but you promote the consumption of highly palatable foods. Okay, so that's consistent with this model of two different types of outputs from the nucleus accumbens. Opioids are promoting approach and reward, and analgesia. Okay, so let's have a model. D2s stop, D1s go. On cells facilitate pain. So that would be aversive. So what my model would be, and I don't know what the connectivity is between the accumbens and the pain modulating circuit, likely through the lateral hypothalamus or the ventral tegmental area, but don't know. So what I'm proposing is you've got the D2s connected to the on cells, connected to the dorsal horn. So this would be your pain facilitating circuit. This would be your pain inhibiting circuit. Okay? This is associated with opioid release. These cells are activated, off cells are activated, and pain transmission cells are inhibited. Okay? This would cause a decrease in pain intensity and increase in pain intensity. So this is how expectations are activating this modulatory system. That was the point of this part of the lecture. Okay. So I think I can summarize now. Pain influences decisions in two ways, both immediately by creating a motivational state, competing with other motivations, and two, by punishing an outcome that results in pain and influencing future behaviors. So this is current, this is future. And that's another thing to really keep in mind. Current, future, bidirectional, facilitation, inhibition. Okay? And it changes your predictions, it changes your expectations. So the avoid pain decision actually enhances pain transmission, paradoxically. And this turns out to be quite important in trying to understand what's going on clinically. Pain signal is a pain predictive cue. an approach reward analgesia system will inhibit pain. So one of the ways that you can deal with pain is create motivations that are conflicting with pain behaviors. Okay, so learning to feel pain, the vicious cycle. Pain predictive cues increase pain. Pain is a pain predictive cue. And pain intensity can increase over time with no change in peripheral input. The way that happens is you have an action and the outcome is worse than predicted. And this maintains and builds the expectation of more pain in the future. Right? So there's a positive feedback loop. And what I'm proposing is that's a normal function of the CNS. That's the learning model. The learning is normal. The behaviors are not helpful to the patient, and they make their pain worse. And the naloxone will interrupt the normal inhibitory effects of reward seeking. So because expectation strongly influences pain intensity, probably the most important thing you can do when you're evaluating a pain patient is to ask them what they expect is going to happen when you do what you're going to do. Right? Don't ask them what they want you to do. Don't ask them what they hope you'll do. Ask them what they actually think you will do. If they're honest, they'll often say, I hope you can help me, but I don't really think you can. And if they say that, the chances that you're going to make those people better are markedly diminished. They don't go away, but they're markedly diminished. So you want to get early intervention. You want to show people that they can get better. You want to change their expectations, and then you can start trying out your medications or whatever you want to do. So right now, I would say the major thing that we would like to find out is what's the actual link between the nucleus accumbens and the pain modulatory circuit, and how does the nociceptive input actually get to the nucleus accumbens? We don't know that. And where the costs and gains are computed, that we have some ideas about. So with that, I want to thank again the committee for inviting me, especially John Levine. I want to thank all the people that have helped. I want to thank Lord Adrian and Pat Wall and all the people that have built this, this wonderful field of pain research. I'd be glad to answer any questions.
So
Right
we have time for a couple of questions.
Yeah.
So one obvious correlate of what you're saying is if patients have treatments that fail, their expectations about all future treatments are going to get worse and worse. So exposing patients to uncertain therapies is possibly one of the worst things that a physician could do. But I wondered if the reverse of that might be true, that if you could make the pain rewarding in some experimental way, whether you'd predict that would lead to pathological pain being suppressed because of the reward, or becoming rewarding in its own right. Does that make sense?
Yeah, I think the answer to that is clearly yes. I think a simpler way to do it would be to actually maybe use experimental pain and model that going away with whatever treatment you have, as opposed to, let's say, the back pain or the chronic headache or whatever they came to you for. So you have to figure out a way to demonstrate to the individual that they actually can get pain relief, because they basically have lost confidence that that can happen. And so there are a lot of patients with chronic pain that should get better with a given treatment but don't. And I believe that part of it is their expectation of relief is very low.
So what do you think would be the role for psychological therapies like cognitive behavioral therapy for changing pain perceptions, because it is used?
Yes, I think they're essential. I think this is a learning process. So it's clear that this is one of the major things you can do with cognitive behavioral therapy is change people's expectations.
So do you use it?
I strongly believe that it would be effective for many people.
Do you use it?
Do I use it?
Yeah.
I did. I used everything. I used acupuncture, lidocaine, opioids, hypnosis.
One last question.
So do you think that the role of expectation has a significant component in the positive effects of acupuncture?
Yes. Yes. I do. I think it also, I think it plays a positive role in every therapy when it works. And I think, John and I did an experiment where we gave people morphine blind or morphine open, right? And at a dose of about 2 milligrams of morphine, the biggest effect was expectation. As you went up in dose to where you got to, say, 12 milligrams, the expectation effect was minimal, and the pharmacological effect was 100%, right? And that makes total sense because it's acting on the same receptors. It's the same pharmacology, right? One is expectation-induced, endogenous opioids at the mu receptor, and one is exogenous. So I think that's a seldom quoted paper that, anyway.
In
whatever.
large literature.
Yes. Okay.
Thank you.